welcome. Today we're going to review the CureGN blood and urine processing kits and procedures. First, you need to ensure that you have the proper kit for your participant population. It should come in one single bag with a urine kit and a blood kit inside. So here's the adult enrollment blood kit, and here's the adult enrollment urine kit. These will be very similar across populations, but they will be dependent upon the participant weight. The first thing that I'm going to show you is what's included in every kit. So in the blood enrollment kit, there are several vacutainers and several cryovials. This can be easily sorted out by pulling out your vacutainers, setting those aside for a moment, and then your cryovials, which you can easily sort by color. So all EDTA process samples will have a red label on them. All SST will have an orange. The sodium citrate will have a turquoise or blue, and then the sodium heparin, which will be light protected by aluminum foil, will also be a dark cryovial. So you could get these set up in advance of your blood draw, which I highly recommend. It's very important to note that there will be 2 blood tubes that will require no processing and that need to be frozen as whole blood. These will both be labeled with a barcode label, whereas all the others will have no labels on them. One of them is the RNA packaging tube, and it will say packaging-RNA, and one of them will be an EDTA blood tube and will say EDTA-DNA. These, again, will require no processing. The next part of the kit is the enrollment urine kit, which will come in a bag that looks like this, and this is a lot more extensive. One thing that I want to note is that whether you have access to cryovial holders or if you just have some vacutainer holders will work just as well. The urine kit is a little bit more extensive and a little bit more involved, but it's also just as easily distinguished by color coding. What you're going to want to pull out first are these 15 ml centrifuge tubes, and one thing that you'll note is that there will be 3 of them that have a red X. These all contain a product called sodium azide, which is just an antimicrobial, but it's preloaded into each of the tubes. And then the other orange capped tubes will have protease inhibitor. So you want to separate these into 2 distinct piles, and then you're also provided a blue capped one, which will be used as a counterbalance for water. So this is just so that you have an even number of centrifuge tubes to spin. For the urine cryovials, there are 5 different types. There is a type for the whole 24-hour urine, timed urine, or first morning collection void, which will all be processed in the same way, and those will each have a brown label on them, which you can set up. The next are 15 cryovials, which will be highlighted in yellow, and they will say Na-N3 for sodium azide, which you can set up. There are a set of cryovials that have no color indicators on them, and those will be for the protease inhibitor, and they will say PI on them. So you'll want to separate these appropriately. There are 2 cryovials that are different than the others, and they are, one is pellet-PI, and that is labeled in a hot pink, and the other is the sodium azide PI, which is labeled in purple. These are for the pellets, which will be the last part of the sample processing, which we'll talk about this morning. So now you'll want to go ahead and get these set up so that they're organized with all the yellow cryovials and all the uncolored cryovials. Okay, now we have separated all of the urine cryovials by color. There will be 15 for the enrollment visit for the adults of sodium azide, Na-N3 in yellow. As a derivative of that process sample, there will be the sodium azide pellet, which is in purple. The next is the protease inhibitor sample, which will have no color label on it, and a derivative of the protease inhibitor sample, the pellet-protease inhibitor PI, which will be highlighted in hot pink. And then finally, for the whole urine sample, will be the brown highlighted cryovial. Okay, now that we have procured our specimens according to your institution's safety guidelines, I want to remind you that the RNA packaging tube and the EDTA DNA blood tube You want to set those aside because those will not be processed. Now we're going to move to the centrifuge, and according to your institution's guidelines, centrifuge all the remaining samples. Next, we want to be sure that we properly balance all of the samples in your centrifuge. So this would be side 1 with the 6 ml SST and the rest 4 and 4.5 ml tubes, and then the 6 ml SST and the 4 and 5 ml tubes here. You also want to be sure that you're providing accurate coverage of your samples. Your bucket should have an aerosol containing device that attaches before you spin. Now that your samples are ready to spin, remember that we're going to spin them for 2000 G for 12 minutes. While the blood is spinning, we're going to set aside the EDTA DNA sample and the PAXgene RNA tube to sit aside. Those need to sit at room temperature for 120 minutes before they're put in the freezer. While we do that, we're going to prepare our urine samples. If your participant brought a 24-hour urine sample, a timed urine sample, or their first morning void, the first thing that we're going to do is weigh it. You can use a simple bathroom scale, or if you have a digital scale, this would work just as well. You're just going to take the entire sample, place it on the scale, and document on your CRF the final weight. I would do this in grams if possible. Next, we're going to measure the volume of the 24-hour timed or first morning sample void. You'll need a container for measuring and your entire sample. Before you start this process, you're going to want to invert your sample gently 3 to 4 times to redistribute any sedimented urine particles. Next, you're going to pour it off and measure it. There's 1,000 mils. And remember for the last one that you have to keep some sample to store. So now we can measure our total volume. It's 3,480. After you've emptied your 24-hour urine collection container, please remember to measure it empty so you can subtract the tare. We measured the total urine and container weight at 3.55 kilograms, which is 3,550 grams, less the 20 grams of the weight of the container. So now what we will store in our CRF is 3,530 grams for the weight of the urine. Here's our 24-hour urine sample processing. Again, this will apply to a timed urine collection and a first morning urine collection only. What we're going to do is take the whole urine sample. We're not going to spin or do any processing to this. We're only going to transfer the urine to the 2 ml aliquots that have a brown label. So once you've identified again the 5 cryovials with the brown label for which the 24-hour urine needs to be transferred, you can either use a digital pipetter or a transfer pipetter, which is just a small plastic disposable one. But what you want to do is bring your sample over, pipette up the prescribed amount for your pipetter or as much as you can hold, and pipette 2 mils approximately into each cryovial. As you'll see from the urine mop and the sample processing mop, the line above the 1.8 is the 2 mil mark, and that's where we're shooting for. Once you've pipetted all of the sample into the 5 cryovials, replace the caps tightly and set aside for freezing. Now that the blood has finally stopped centrifuging, we're going to carefully remove the samples so that we don't disturb the centrifuged portions. So we're going to lift them out. This was our water balance. And you'll notice that they've separated into blood and plasma. So carefully remove all the samples. As can be found in your manual procedures for specimen processing, we've created a short guide for processing the blood specimens. Here we have all the blood tubes that will potentially be drawn depending on your participant population, as well as the EDTA DNA tube and the PAX-gene RNA tube, which we do not process. But those that we process, again, we've spun them for 12 minutes at 2000 G, and we now have processed samples. Processed samples which will go into color-coded corresponding cryovials. Okay, to transfer the blood plasma and serum into the corresponding cryovials, it's important to remember that the sodium heparin, light-sensitive one, can be a little bit difficult to see. So what we have done here is taken a razor blade and made a slight incision into the foil, which will not damage the sample at all, but this just makes it easier for you to use a pipette or a pipetter to retrieve the sample. Now that we've removed all the caps from our vacutainers, we're ready to transfer, except for the sodium heparin. It's fine. We're ready to transfer the plasma and serum over to the cryovials. If you're using a digital pipetter, you'll want to set your volume to 1 mL so that we can transfer over 2 mL into each type. We recommend that you use a filtered pipette tip for blood specimens, and as required by your local institution, you'll want to use either plastic protection device or a hood, which is to prevent any spray from biological materials. Remember that you're going to want to change your tip between sample types. For example, as we do the sodium citrate, and we transfer this over to the cryovial, we have 1 cryovial for 2 mL of sample. After we've completed that, we're going to exchange tips and get a new tip for the sodium citrate sample. And again with the EDTA. You're going to want to fill all cryovials to approximately the 2 mL mark if you're using a transfer pipetter. And once they're all filled, reattach the cap tightly, and they're ready to be frozen. Now that we've finished transferring all of the plasma and serum into the cryovials, we want to recap our used vacutainers and dispose of them properly in your institution's biohazard bin. As part of the NIDDK shipper kit that you should receive directly from the NIDDK biorepository, there should be some freezer boxes. Now would be a good time to transfer your plasma and serum samples over to the freezer box. Filling from the top left corner across. One thing I want to mention is that if your blood draw was not enough to fill all the cryovials, you're going to want to keep that cryovial aside as an empty container, and then you'll have to indicate that on the CRF at a later time. Now we're going to start with the urine processing for the spot urine collection that should have been collected at the study visit the day of. First, you're going to separate the 15 milliliter centrifuge tubes by the red X's, which have sodium azide in them, and the unmarked ones, which are preloaded with protease inhibitor, or PI for short. Also, you're provided one additional one that's going to be used as a counterbalance for the centrifuge, which should have a blue cap and will be filled with water. These are all going to be spun for 12 minutes at 1000 G. You're going to have your spot urine sample, and the goal here is to collect at least 70 cc's. However, 75 would probably be a little bit better. Just give that a gentle inversion, and next you will pour it into your samples containers. Next, we're going to fill all of the 15 milliliter centrifuge tubes with 10 cc's of urine. And repeat for the PI tubes. Now that you have completely loaded the 15 ml centrifuge tubes with the spot urine sample, you should also have prepared your blue cap water balance weight, and we're going to take these over to the centrifuge to spin. Taking care to balance your centrifuge carefully and using your institution's approved covers for biological specimens, you're ready to begin spinning. Now that your centrifuge is ready to spin at 1000 G for 12 minutes, we can go over and take a look at the rest of the supplies that should have been provided at your initial kit delivered. One thing that you should have received in your initial shipment of materials for the study is a 15 milliliter centrifuge tube filled with about 5 milliliters of RNA later. This is needed for the pellet processing of the urine samples and should last you approximately 1 year. After that, you will get an installment of the next 5 mils, which we ask you to keep for the duration of the study. Now that our spot urine sample has been spun down, if you look carefully, you should be able to see a small pellet at the bottom of the tip. Take care to not disturb the pellet. To transfer sample from the 15 ml centrifuge tubes to the 2 ml cryovials, please remember that from those centrifuge tubes marked with the red X, we're going to be using the yellow labeled NA-N3 cryovials. For the enrollment visit of the adult participant, this will be 15 cryovials. These 30 mils of sodium azide mixed spot urine sample should fill the 15 cryovials, which are 2 mils each. So you should essentially use all of the supernatant sample from each of these centrifuge tubes. So you're simply going to transfer over 2 mils into every cryovial. And you'll continue to do that until you've used up all of the sample. From the centrifuge tubes that are not marked, we're going to put the sample into the unmarked 2 ml cryovial tubes, of which for the PI or protease inhibitor sample type, there should be 20 cryovials. So there are 40 mils of urine that have been spun that will go into 20 2 ml cryovials. Again, you're just going to transfer over 2 mils of urine into each cryovial. Now that we've transferred all of the supernatant into the sodium azide and the protease inhibitor cryovials, we should be left with just the pellet. There may be a small amount in the bottom of your centrifuge tube, for which I would like you to invert that for 10 seconds onto a clean, dry paper towel. And then you'll be left with just the pellet. You should do this for all 7 centrifuge tubes. Now that we've removed all of the possible supernatant from our centrifuge tubes, we're going to open up the PI pellet cryovial, which is in hot pink. There should just be one, and the NA-N3 pellet cryovial, which is in purple, which should also be just one. So what we're going to do now is take the RNA later that we received as part of the initial shipment, and we're going to transfer over 20 to 50 microliters into each centrifuge tube to cover the pellet. If you have a transfer pipetter, it is fine to just use 1 drop of RNA later. The added RNA later to each of the tubes, you can simply tap any that is on the wall of the tube down and flick the bottom of the centrifuge tube with your finger. You do not need to pipette up and down to mix the pellet with the RNA later. Next, we're going to transfer each of the 3 sodium azide pellets, the NaN3, those marked with the X, into the purple labeled NaN3 pellet cryovial. All 3 pellets can be aspirated and put into the single cryovial. Changing tips, you can do the same thing, or getting a new transfer pipetter with the PI sample into the pellet-PI cryovial, which is labeled in hot pink. Once all of the 4 centrifuge tubes are transferred over into the hot pink cryovial, that is the end of the urine processing. After you've completed the urine processing, remember to store them with the blood processing samples in the freezer tube provided by the NIDDK biorepository. If you did not use any cryovials because your participant did not give a 70 cc urine sample, remember to set those aside so that those can be designated as not used in the CureGN link CRF. Please remember to dispose of everything properly according to your institutional guidelines and put the samples in the -80 freezer promptly. The RNA PAX gene and the EDTA DNA blood sample need to sit at room temp for 120 minutes or 2 hours and then can be placed in the -80 freezer. Thank you for your attention. If you have any further questions about processing, please refer to the urine and blood processing MOPS in the Manual of Procedures, or contact me directly via the CureGN monitoring at arborresearch.org email address. Thank you.